Now let's study necrosis. So necrosis is defined as a sum of the morphological changes indicative of cell death in a living tissue. It is caused by the progressive degradative action of enzymes following harmful injury. It is an accidental and unregulated form of cell death. Okay, it results from damage to the cell membranes and loss of ion homeostasis. So severe injury causes a rapid and uncontrollable form of death, and it is called accidental cell death, and the morphological manifestations of the severe damage is called necrosis. It occurs accidentally. Okay, so the necrotic cells cannot maintain integrity of the membrane, and their contents, including the cellular enzymes, they leak out and ultimately digest the cell. This brings out acute inflammatory reaction. So the enzymes which digest the cell are either derived from the lysosomes of the dying cells themselves or from the leukocytes recruited as part of the inflammatory reaction. For morphology, you can see cytoplasmic changes. So remember, everything in necrosis is M. Okay, in cytoplasm you can see moth-eaten appearance because the cytoplasm becomes vacuolated because it is being digested by the cytoplasmic organelles. So here you can see moth-eaten appearance and increased eosinophilia, homogeneous eosinophilia. Next, nuclear changes, you can remember it as pycnosis, karyarexis, and karyolysis. Pycnosis is shrinkage of the nucleus, which appears shrunken and deeply basophilic. It appears like an ink drop. Here you can see a pycnotic nucleus. Next, karyarexis is breakdown of the pycnotic nucleus into smaller fragments. Okay, and karyolysis, there is lysis. So nucleus is basophilic, right? So there is loss of that basophilic staining of the nuclei because of digestion of the DNA by DNA's activity, and it leads to a ghost nucleus. In 1 to 2 days, the nucleus of dead cell may completely disappear. So cytoplasm, moth-eaten cytoplasm with homogeneous appearance, and also pycnosis, karyarexis, and karyolysis. Electron microscopic findings, you can remember it like this: MMM. So there is MEM for membrane discontinuity. So lysosomal disruption is there. They release their enzymes, and that causes the digestion of the cell. Next, there is marked dilation of the marked dilation of the organelles, and also there are large amorphous densities. So mitochondrial densities, marked dilation of the cell, membrane discontinuity, and myelin figures. So presence of intracytoplasmic myelin figures is there. So fates of necrotic cells. After necrosis, these necrotic cells may persist for some time, or they may be digested by the enzymes and they disappear. So dead cells may be replaced by myelin figures, which may either be phagocytosed by other cells or may undergo degradation into fatty acids, and these fatty acids can bind calcium salts and the necrotic cells may become calcified. Okay, so moving on to the morphological patterns of tissue necrosis. First, we have coagulative necrosis. So in coagulative necrosis, what happens is there is coagulation. So ischemia denatures and coagulates the structural proteins and enzymes. So this blocks the proteolysis of the dead cells. So because the proteins are still there in the same structure, the structure of the cell is maintained. So the outline of the dead tissues is preserved, and also infarct is a localized area of coagulative ischemic necrosis. So ischemia is caused by obstruction in a vessel. So mechanism: ischemia denatures and coagulates the structural proteins and enzymes. This blocks the proteolysis. So organs affected. So organs with end arteries are more affected, but brain is not at all affected by coagulative necrosis. You only see liquefactive necrosis, CNS. Okay, so end arteries you have the kidney, heart, spleen, etc. And limb also you can see dry gangrene. Now, the appearance of necrotic tissue is described as coagulative necrosis. Okay, it describes the coagulation of proteins that occurs upon heating. So you can see the involved region appears dry, pale, yellow, and firm. So pale in fact, right, in spleen. So dry, pale, yellow, and firm. It is wedge-shaped in organs like the kidney and spleen. So microscopically, the normal cells are converted into their tombstones. The outlines of the cells are maintained, but their cytoplasmic and nuclear details are lost. So because karyarexis, karyolysis, all that cytoplasmic and nuclear details are lost. So that is why in kidney you can see here the tubule structure is maintained, the outline is maintained, but the nuclei are lost. See, the cells don't have nuclei. And also nucleus may be absent or show karyolysis. Leukocytes are recruited to the site of necrosis, and the lysosomal enzymes of the leukocytes, they digest the necrotic cells. The resulting cellular debris then is removed by phagocytosis by infiltrating neutrophils and macrophages. Next, liquefactive necrosis is also called co-liquative necrosis. So there is liquefaction. Why means because the enzymes are digesting everything. The enzymes of the leukocytes and lysosomes, they digest everything. Infections by bacteria also, they stimulate the accumulation of these enzymes. They digest or liquefy the tissue. So liquefaction is due to digestive action of the hydrolytic enzymes released from the dead cells and leukocytes, both autolysis and heterolysis. So the dead tissue rapidly undergoes softening and transforms into a liquid viscous mass. So causes are ischemic injury to CNS and suppurative infections. So moving on to gross morphology, the necrotic cells are completely digested and converted into a viscous liquid that is removed by the phagocytes. Okay, the involved area appears soft with liquefied center. The organs affected are brain. The necrotic area is soft and center shows liquefaction. And because there are abundant lysosomal enzymes in the brain or CNS specific hydrolysis. And then the bacterial infection causes acute inflammatory reaction and produces liquid viscous material called pus. The localized collection of pus is called abscess. There could be abscess anywhere. And it's also seen in wet gangrene. Ischemic coagulative necrosis is seen in dry gangrene, and wet gangrene you can see liquefactive necrosis. Next, microscopy, you can see the pus, which consists of liquefied necrotic cell debris and also dead leukocytes and macrophages. Caseous necrosis, it is seen in tuberculosis. It is characteristic and due to type 4 hypersensitivity reaction. It shows combined features of both coagulative and liquefactive necrosis. So basically, organs affected are lung and lymph node in TB, though it can involve any organ. And appearance, see caseous, cheesy, yellow-white, soft, friable, granular appearance. So if you look at cheese, it is granular, right? So Swiss cheese especially, it is yellow-white, it is soft, it is cheesy. So that is the gross appearance. Microscopically, the focal lesion of tuberculosis infection is granuloma. Caseous necrosis is the collection of fragmented or lyse necrotic cells that appears to be eosinophilic. You can see pink here, granular material, cheesy white, all that, right? So there is complete obliteration of the tissue architecture, and that is in contrast to coagulative necrosis. There is obliteration of the outline of the tissue. It is not possible to identify the cellular outlines. It is surrounded by epithelioid cells, Langhans giant cells, and fibroblasts forming a granuloma. The caseous necrotic material may undergo dystrophic calcification. Fat necrosis. So it refers to focal areas of fat destruction. So enzymatic fat necrosis can occur in acute pancreatitis. So the enzymes from the SNR cells, they leak out into the substance of the pancreas and peritoneal cavity, and they lyse the tissue. They cause tissue damage. They destroy the fat cells, and the free fatty acids are liberated, which combine with calcium and form calcium soaps. This is called fat saponification. So morphology, it appears as chalky white areas. Here you can see chalky white areas grows in the omentum, which helps them to identify the lesions. And microscopically, the necrotic fat cells, they appear pale. Okay, the cells appear pale with shadowy outlines surrounded by calcium deposits because of calcium saponification and an inflammatory reaction. Traumatic fat necrosis occurs in tissues with high fat content like the thigh, the breast, etc. following severe trauma. Next, fibrinoid necrosis is characterized by deposition of pink staining fibrin-like proteinaceous material in the tissue matrix in the vascular vessel wall with staining pattern reminiscent of fibrin. It obscures the underlying cellular detail, usually seen in immune-mediated deposition of antibody-antigen complexes in the walls of the vessels. So you can see it in vasculitis, for example, polyarteritis nodosa pan, malignant hypertension, Ascough bodies in rheumatic heart disease, and preeclampsia placenta and hyperacute transplant rejection. So you can see eosinophilic hyaline deposit in the vessel wall. You can see it better with phosphotungistic acid hematoxylin stains or PETA stain. So in the vessel wall, you can see fibrinoid necrosis, glassy eosinophilic, bright eosinophilic hyaline-like deposit. Gangrene. So gangrene is massive necrosis with super added putrefaction. This term is important. So two types are there, dry and wet gangrene. So a variant of wet gangrene known as gas gangrene. It is wet gangrene. It is caused by Clostridia. So dry gangrene, it is caused by arterial occlusion, usually involves a limb. So grossly, you can see a darkness, obviously, and also you can see dry, shrunken and shriveled appearance. The foot especially resembles foot of a mummy. The black color is due to iron sulfide, FeS, and there is a line of demarcation which is seen between the gangrenous and the normal area. Microscopically, the necrosis is of coagulative type. It shows smudging of soft tissue. There is smudging of soft tissue, and also the line of demarcation consists of granulation tissue with inflammatory cells. Wet gangrene, it is caused by venous blockade because of hernia intussusception or volvulus. Diabetic patients may develop both dry and wet gangrene. And sites, it occurs in moist tissues or organs. So the affected part is swollen, putrid and dark here, but instead of shrunken and shriveled as in dry gangrene, it is swollen and soft in wet gangrene and superadded putrefaction. Microscopy, you can see liquefactive necrosis. Gas gangrene is a special type of wet gangrene caused by infection with Clostridium perfringens. They enter into the tissues through contaminated wounds, and toxins cause local necrosis, edema, and also they are absorbed. So gummatous necrosis, it is seen in syphilis. The necrotic tissue is rubbery gum. Okay. Lab detection. So the intracellular enzymes, they leak out when there is necrosis. So cardiac muscle, you can see creatinine kinase and troponin. Hepatic bile duct epithelium, you can see ALP, alkaline phosphatase, and also hepatocytes, transaminases. And finally, differences between dry and wet gangrene. Dry gangrene in the limbs. Here it is in the bowels. So we know the examples. And again, etiological factors here: arterial obstruction here, venous obstruction. The rate of obstruction in arteries is slow. And wet gangrene, because of bowel volvulus, it is rapid or abrupt. The appearance, it is shriveled and dry and swollen and soft. Line of demarcation is present in dry gangrene. The spread is rapid in wet gangrene, and the prognosis is poor due to severe septicemia and wet gangrene. Phoneus gangrene is seen in scrotal skin.